publicized by St. Georgi, one of the men who was instrumental in working out scurvy and some other vitamin deficiency conditions. It was very well publicized. Paprika, for instance, it happens to be that St. Georgi is, I believe, a Hungarian. At least he comes from the south of Europe, where paprika is commonly put in Hungarian goulash and such things as this. And whether that was the reason or not, there was a great splurge of interest in putting paprika in foods quite many years ago when this was first worked on. Now we call them bioflavonoids. I'm quite sure they are not vitamins in the sense that they're absolutely required in the diet, but there's still surges of interest, and there's where, on the basis of where there's smoke, there's fire. There does still seem to be some nutritional value to a flavonoid content in the diet. Currently, the greatest interest is in Russia, and you'll read quite a number, well, you read abstracts of quite a number of Russian articles on the importance of flavonoids in diets. And interestingly enough, wine is quite high in bioflavonoid content, particularly red wine, so that it is being touted in Russia as a way of avoiding this kind of deficiency. Whether there's anything to it or not, one doesn't really know, although I will say one of the healthiest 80-year-olds I know eats extract of orange peel a couple of times a week, and maybe that has something to do with the fact that he's so healthy. At least it doesn't seem to be hurting him any. And he also drinks a little red wine, too. The flavor values we've already illustrated by examples, and on page 2 of these notes you have some examples of things that occur in natural foods as important aspects of flavor. Thymol and thyme and thymol and thyme, excuse me, I got the THs backward, and other components including clove oil, but in much smaller amounts there. Anethole is a component of anise flavor, has a rather nice anise odor. Safrole, the nice sassafras odor. Cinnamates in cinnamon. Cresol and other compounds in smoke flavors. Chavicol has been reported in pineapple. I personally doubt that it's an important component of the pineapple flavor, but maybe that's personal prejudice since I didn't find it and others did. Eugenol, certainly a major component of clove oils and a few other things. Guaiacol in medicinal odors in some natural foods, medicinal preparations in some natural foods, and smokes as well, and so on. The galangan and zingerone and so on in ginger are important to their flavor, and phenol itself is found in summer savory, which is used as a flavoring ingredient. So basically phenols have several kinds of flavor effects. They can have, if they have sufficient volatility, they can contribute to odor, and they can be involved in the indirect production of odorants. So that, for instance, the Strucker degradation of amino acids is a reaction that involves first the formation of a Schiff's base between the amino acid and a carbonyl compound, which very likely would be an oxidized phenol in the natural system. And then the reaction proceeds to remove the amino group, leaving behind a keto, and the alpha keto acids are easily decarboxylated. The result being that instead of amino acid, you'd have the carbonyl compound one less carbon long. So that, for instance, from phenylalanine, you'd get phenyl acetaldehyde, and phenyl acetaldehyde and its reduced product phenyl ethyl alcohol are very rosy compounds. So these would be examples of how indirectly the phenols may help produce odorous compounds. And this is thought to be quite important in some aspects of black tea odor, and it's been postulated as important in some aspects of aged bottle-fermented champagne odor, for instance. I think these reactions are less well demonstrated there than they are, for instance, in certain kinds of baking products and in heat-treated or heavily oxidized materials like the black tea. Bitterness, of course, can be related to phenols, and we'll spend a little more time on that, but it's certainly true that lots of bitter substances of natural foods are not phenols. In fact, the most bitter substances like quinine are usually alkaloids, and some of them are phenolic, of course, but need not be, the significant feature of the alkaloid being the nitrogen component. Other bitters include certain glycosides and terpenes that are not phenolic either, so that probably the most important bitters in the really objectionable sense are not phenolic, but the mild bitterness that is acceptable and even desirable in some foods, and particularly in wine, does seem to be phenolic. Astringency is extremely important related to phenols, and as far as we know, there are no astringents in foods that are not phenols. Now, there are a couple of possible exceptions to this. You may know of, I guess none of you are old enough to be well acquainted with what was called a styptic pencil, but they used to, when you shaved with a straight razor, you hacked yourself pretty regularly, and then you had a little white pencil that you'd touch and the blood would stop. Well, the astringent effect to stop the blood, and also if you put it in your mouth, the astringent flavor, was due to alum, which is aluminum sulfate, and aluminum sulfate and a few other mineral compounds like that are somewhat astringent, but they don't occur in foods in any normal amount, normal degree sufficient to give this kind of astringency. And the only, so that while it might be possible, we don't know of any examples of significance in normal foods or natural foods. The other group of compounds that have recently been discovered and claimed to be astringent are certain peptides from cheeses, so that in cheeses, bitter, cheddar cheese in particular, bitter and astringent components of the flavor are reported to be due to certain small peptides. And this field is quite active right now. You may have heard of aspartame, which is a sweet peptide, which has recently been approved for addition to foods, and will be showing up instead of cyclamate and so forth in foods as soon as the further details are worked out. So we can't certainly say that all astringency is invariably due to phenols, but certainly all the important known cases, and again in wine, the phenols are the sole source of astringency of any magnitude. Of course, there are other components to flavor. There's mouthfeel, and lignin is perhaps important in mouthfeel, certainly in the fibrousness and the texture, but I think it's important in other aspects of mouthfeel. For instance, oak contributes to wine two kinds of flavor from my viewpoint. Well, maybe three if you count astringency, although generally it should not be present at a sufficient high level to be noticeable in astringency. But it contributes an oak odor, and American oak is very high in this oak odor aspect, but it also can contribute what I describe as a rich flavor. And particularly old conditioned European barrels are, and old barrels in general are good in this contribution of richness. And in trying to describe the richness, it does contribute to apparently mouthfeel, possibly vanillin and some similar derivatives are involved. There is a small odor component, but I think what the main contributor is the small alcohol-soluble lignin degradation products that do appear with time, and there are some amounts present in oak and other woods even without the reaction time with the beverage. If you react lignin in alcoholic solution, you can fragment it slowly, particularly under oxidative conditions, and liberate phenolic material in the form of lignin products. And I believe this is what is contributing this rich flavor. That's fairly inferential without very much good experimental data upon it, but it is true that if you put soluble lignin in an aqueous solution, it certainly does change the feeling in the mouth and some aspects of flavor. The texture value is fairly obvious. Lignin is associated with fibrous tissue, even in the plants that we do eat, and there is some astringency relation because the lignin is associated with the vascular strands, you see, in a fairly soft flesh material, and so is the tannin. So that generally speaking, the astringency and lignin do tend to go hand in hand, and the more fibrous vegetable also is likely to be more astringent because of the physiological association of the two kinds of compounds. There are Christmas aspects And the turgor of the cell has a lot to do with crispness. If we speak of a crisp grape, we mean a turgid grape that pops when you bite it and has a mild resistance and a crisp feeling when you chew it up. But the same kind of effect is found in celery and asparagus and beans, rhubarb, and so forth because of ratios of lignin relationships. So actually, the more crisp and the less tough would be the one with the lowest lignin in most cases, but at least there is a lignin relationship and possibly a water holding and other phenol relationship as well, although the phenol content of the preferable condition of celery, asparagus, beans, or rhubarb is quite low. Lignification in peas is of a more general and less fibrous type, but you all know what a overripe pea tastes like. It's very hard and unappetizing, and you have to chew pretty hard to eat it, and that is due to lignin increase, even though it isn't recognizable as a fibrous condition. Pears is a rather interesting example. Pears and a few other fruits contain stone cells, and these so-called stone cells are rather you know the gritty feeling of the pear flesh in your mouth. These are rather isolated centers of lignification, and the ideal pear has few stone cells and mostly non-lignified flesh. There are pears that are like eating wood that you just couldn't really eat, that are completely lignified, and the proper distribution of minimal amount of lignin has something to do with the texture and mouthfeel, if not flavor value, of the pears. Color value is just fairly obvious, and we'll spend a good deal of the remainder of the class on anthocyanin color and its desirability and changes, and then the browning colors and their formation and off colors, so that we needn't spend any time on it here. And then there are what might be called product aspects. That is, the raw product can have phenol stories related to growing, harvesting, or storing, so that we either obtain or retain the correct phenol relationship. So, for example, the pear softens partly by pectin hydrolysis and becomes ripe after being picked if the conditions are correct, but also the amount of lignin formation and how prominent the stone cells are also can change after the fruit is picked and may have a bearing on quality, as is true of string beans and asparagus and so on. And true of grapes too, of course, and ordinarily we would want to pick the grapes at a time when the phenol content is about its maximum for other reasons, for winemaking in terms of getting the sugar right and so forth, but incidentally it also gives the proper picture to the red wine and some aspects to white wine. Then there are processing effects. The enzyme effects are fairly obvious from wine, and there can be effects of heat or oxidation, some good and some bad. For instance, pears sometimes turn pink in the can. If you can them, the heat treatment makes them turn pink. This can be rather attractive. The same thing may happen to applesauce, but it depends on what the can's made of and so on. The pink may turn to blue or a dirty gray color, and that's certainly not attractive. And this is a leukoanthocyanin type change, and there are a number of such examples where you either want a change or you don't want a change depending on the situation. One of my favorite products that's very poorly known and deserves to be better known, I think, is persimmon pudding. And it can be made from the Japanese type persimmons or the Midwestern type of persimmons. And the persimmon, as you may know, is a nice bright orange fleshy fruit when sufficiently ripe, soft tissue, and not very tannic, but terrible if it's underripe. And the persimmon pudding is very brown, looks browner than apple butter, if you know what apple butter looks like. It's brown as my coat or so. And this is partly then the flavor is due to the tying up of the tannins in an oxidative state, and it makes a very attractive dish served with lots of heavy cream, and I can just visualize it right now. The storage changes can be important. We've mentioned browning already, and the oxidation effect and possible antioxidant interaction, and certainly antioxidants added to fats are nearly all phenolic. Many of them are originally natural sources, The microbial effects we've hinted at. Sometimes the spoilage can be prevented or is minimized by the presence of phenols. We've mentioned onions as an example as to why the white onions are more subject to damage in storage than the yellow or red ones due to the antibiotic effect of phenols in the outer layers and so forth. Precipitations, hazes, turbidities in beer, wine, and other products are phenol related. And then there are effects related to the container, interaction with light, the oak barrel, for example, extraction, metal content that interact with the phenols. I think we mentioned one example of pork and beans where the pork turned green, and the reason it turned green is that the pork and beans had been cooked in a copper container, and the quercetin of the beans and the copper combined to make a blue-green pigment which collected in the fat of the pork and made it look very unattractive. So this is the examples of that kind of thing. And then there are certain products that have very important relationships to phenols. Fruits, wines, and related products like ciders and beer we've already either mentioned several times or hinted at. Brandy and whiskey are important from the barrel extraction viewpoint. Tobacco has a very interesting story related to phenols in that the browning reactions are phenolic and the curing mechanisms depend on phenol reactions, and the odorous products to some extent are the result of changes resulting from the phenol oxidation during its curing process. Smoking of meats and such things contributes smoke phenols, which are important flavor constituents. Tea is almost completely a phenol story, and other teas too, like mate, which is a South American tea, and what's called Mormon tea in Utah and there's a rooibos tea, which I learned to like very well in South Africa. It's not exported in any great amount, but is an interesting tea, which has a high, it's a legume, I think it has a high coumarin content and is very pleasant flavored. It may be carcinogenic, so I don't know whether I should advertise it or not, but at least it's very tasty. Coffee has chlorogenic acid in considerable amount, and so on. Spices and flavorings, particularly vanilla, nutmeg, and cinnamon, we've already mentioned several times. And cocoa and chocolate have important phenol stories. The reason chocolate is black, or dark brown rather, is because of phenol oxidation. And a good deal of the flavor of chocolate is because the phenols have been rather completely oxidized and then ground up very finely, so finely that they actually are molecularly broken. That is, the fine grinding will break the larger tannin molecules down, and a good deal of the flavor and mouthfeel of chocolate has to do with a happy combination of the fat of the chocolate and the finely dispersed phenol of the oxidized form. And a lot more needs to be known in some of these areas, but a good deal is known, enough at least to whet our appetites for further knowledge. Going ahead with phenols as flavors, we'll go rather quickly through the examples that don't relate directly to wine. Phenols as odors, you've had examples in the sniff bottles passed around last time. Vanillin is the major odorous constituent in vanilla, although not the only one, but can be the only one in synthetic vanilla extract. It has a threshold about 10 to 25 milligrams per liter, depending on what you're putting it into, and they only put around 20 milligrams per pound in such foods as cake and other things that are vanilla flavored, or maybe ice cream. Ice cream maybe takes a little more, since being cold it's a little harder to smell, but it's a rather odorous material and certainly very desirable flavor. The smoke constituents are mentioned, and notice that the thresholds in water are extremely small, down to a couple of hundred parts per billion, and usually not over 1 part per million, and therefore it doesn't take very much to flavor bourbon whiskey or smoked ham or what have you. Phenol has been found, and chlorophenol has been found as contaminant in beer, probably from water treatment, or sometimes from, I've had an experience in South Africa when we were drawing all of our water from a reservoir. In the fall, all the leaves from the trees fell in the reservoir. And even though the water was deionized, it didn't remove the phenols, so I had to redistill all the water from alkaline solution to avoid getting so much phenol in my dilution water that I couldn't do phenol analyses. So that water that may come from an open reservoir and have phenol from leaf extract or other possible source of contamination when used to make beer can give contaminants sufficiently strong to give abnormal flavors. And these abnormal flavors, again, have been found at the order of a few tens of parts per billion, so that they can be very obnoxious at anything over that level. The tyrosol content does give a phenolic type smell and a bitter taste in aqueous ethanol, and does occur in fermented beverages by yeast action on tyrosine, or yeast production from carbon units related to tyrosine, so that it probably does contribute in beverages. The amount found in beverages has been as high as 40 milligrams per liter, so that clearly if the threshold is around 14, then it must have an effect, or can have an effect at the level present in some fermented beverages. Clove Bud Oil is mostly eugenol, thyme oil is mostly thymol, and sassafras oil is nearly 100%, or can be, saffrole, and so on. Mace is about the same as nutmeg oil with its myristicin content and some saffrole. Cinnamon oil is rather high in cinnamaldehyde, and in fact that's how cinnamaldehyde got its name. And cinnamic acid as a derivative gets their name from cinnamon. Wintergreen oil is not the wintergreen leaf is not odorous as picked, because the wintergreen is present. The wintergreen oil, methyl salicylate, is present as a sugar glycoside, but it is fermented, as they would say, as tea is fermented. That is, the leaves are allowed to wither and incubate without the enzyme being destroyed, and the enzyme converts the glycoside to the aglycone, and then the odor develops. This is not used anymore, or never was used commercially as far as I know, but the Indians and the French in the Quebec eastern area used wintergreen tea as an important part of their condiments, at least in the early colonial and pre-colonial period. Phenols can be pungents in addition to being odorants, and the pungent character is not limited to phenols again, but some of the best known are phenolic. And what do we mean by a pungent? Well, we'd mean those things when put in the mouth give a penetrating burning effect, and sometimes can be recognized as pungent in odor. The flavor in smoke that's called empyromatic is sort of a cough-producing effect, and is part of the burning aspect of smoke, and it does relate to the phenol content, apparently. Capsaicin, a rather interesting amide, and it's drawn wrong in your notes unless I've corrected it. It is a carboxyamide, so the strange-looking oxygen with the carbonyl attached is really a carbon. So this compound is the hot pepper effect in cayenne pepper, and it takes very little to make the pepper very hot. And you can assay them for hotness by assaying for capsaicin content. Clove oil, you may have had, if you're as old as I am, you've probably had a toothache treated with clove oil, where the clove oil, you put the clove oil or a clove even, on your gum near the tooth, and it is a sort of a counter-irritant. It makes a hot feeling in the mouth, and this takes your mind off the toothache. I don't think it helps it any, but at least it takes your mind off of it. So this kind of thing can be significant in flavor of cloves and ginger. Ginger is pretty hot, and the hotness is due to zingerone and related compounds in it. Ferulic acid and its ethyl ester have been reported to give a pungent taste when added to sake at only 2 to 3 milligrams per liter. And theaflavin, which we'll talk about, I hope, later on if we have time, is a phenol derivative that occurs in black tea, and it contributes brisk flavor to tea. And you've heard all the tea ads with commenting on briskness. and this is related to, if not completely due to, the theaflavin content, and probably a pungency effect. And this then brings us to astringency, where the phenols really shine. Now, any phenol that's large enough to have a molecular weight of the order of 500 or bigger is astringent. And the word astringent derives not only, or we think of it as a flavor component word, but it was originally used, or is additionally used, in connection with tanning of hides. If you put a piece of animal skin, human skin for that matter, into a tannin solution, and this piece of skin measures 4 inches square, after it's tanned it'll measure 3 inches square or something smaller. So the shrinking that's produced by the tanning process is related to the astringency. And it's been known for a long, long time by the tanners that they could taste the solution, and the astringent taste was related to its efficiency as a tanning solution, and that in turn to the amount of shrinkage produced, so that the most shrinkage gives the best tanning. And this was shown to be, after more modern techniques, that as far as tanning leather, you must have a molecular weight between about 3,500 and maybe, well, no, let's see, I guess it goes a bit higher, say between about 5,000 and about 1,000. If it's below 1,000, it'll still taste astringent, but the molecules will be too small and they'll leach out of the leather very readily when the leather gets wet. If they're much larger than 5,000, they won't get into the leather, and if they will produce what's called a case hardening effect. So the skin is tan on the outside, but the inside isn't, so that if microbes get in, it'll rot from the inside, and it'll be like spoiled meat on the inside and tan on the outside, and of course that's not very happy from a shoemaker's viewpoint. So that the molecular size is related to astringency, and the smallest molecules are known to be less astringent or non-astringent if they're sufficiently small, and the larger are tasteless or non-astringent in the tannin shrinkage, leather shrinking sense, apparently because they just don't interact very well with the taste buds, or with the, can't penetrate the hide. This has been pretty well shown to be almost exclusively, if not exclusively, related to hydrogen bonding. And the hydrogen bonding, of course, is with the phenolic OHs. So the sugars, regardless of the OH content, would not be astringent because they don't hydrogen bond appreciably. They're not acidic. But the acidic phenolic hydrogens do hydrogen bond rather strongly, and this produces the effect of astringency. Now, astringency is related to a number of things, and we don't entirely know the exact mechanism, but it's partly related to the actual combination of tannin and protein on the mucous membrane. And if you've been on a wine panel and noticed that after you spit out the red wine, the saliva is coagulated and the protein is certainly precipitated by the tannin, then you notice your mouth feels differently. It feels slicker in some places and rougher in other places because the mucoproteins have been removed. So that's one aspect to astringent flavor, but there seems to be more to it than that. If you take some really astringent material into your mouth, it has a grabbing effect that must be some response more than this. And the only work I know of that hints as to what this might be is a small report in Joslin and Goldstein's report on astringency and advances in food chemistry, which I believe is, yes, is number 6 on your reference list. In this paper, they suggested that this may be due to catecholamine interaction, pointing out that, of course, catecholamines are inactivated by methylation. And if you could tie up methyl groups, then you would prevent their inactivation. Since the catecholamines are nerve messenger compounds, certainly you would get a tingle or an otherwise nerve-type response that might be pretty outstanding. And they based their opinion on the fact that if they rinsed their mouth after a very potent astringent compound with methionine, then it lowered the lingering effects of the astringency. And it does work. I've tried it. You can try it too. Unfortunately, methionine has a kind of a, generally as you buy it anyway, sort of a sulfurous skunkiness, and therefore it does contribute other problems. I've never tried it with choline, which might not have that particular effect. But in any case, there does seem to be a nerve-ending interaction that probably is related to some effects with the catecholamines. Bitterness is a separate entity, and bitterness, of course, is a flavor, one of the primary tastes, and astringency is not. But they are so closely related in that high tannin content also tends to go with high bitterness, and tasters know this, so they have a tendency to be difficult to convince that if something is, if you're trying to get them to rate things separately for astringency and bitterness, you may find some difficulty. Even I think, I've never tried this, but I'll bet you if you ask people to rate white wines that had quinine added, I bet you that they might raise the bitterness rating a lot, but I bet they would rate the astringency rating as slightly higher if you'd added quinine, and there would be no added astringency from that. This makes it difficult to study the two things separately, but by careful admonition and training of your panel, you can do some work in this regard. The tannic acid threshold, tannic acid added to water and to peaches runs about 200 milligrams per liter in the peach or in water, and then the minimum detectable difference was around 400 milligrams per liter. In other words, if you add some tannic acid to water so that you can taste that it has tannic acid in it, then add more and ask people to rate whether or not it's at an increased level. The difference threshold is higher, of course, than the initial detectability threshold. And these sorts of figures are interesting. And if they add the tannic acid to peaches, they get about the same order of difference, in that 150 to 200 milligrams per kilogram of peach was detectable. And if you measure the natural tannin, it took much less, so that in peaches that had 151 versus another group that had 181, 6 out of 12 judges could detect the additional tannin when that was only 30 parts per million. So the distribution has some bearing as well, perhaps, and maybe the kind of tannin. With grapeseed tannin, a number of studies have been made, and the earlier studies found rather low thresholds, but by using more purified phenols, we've done a better job. Not that the old work was bad, but at least by having more purified samples, you can get a much lower threshold. Excuse me, that isn't the particular one I wanted to show first. Let's do this one first. Thresholds in water for grapeseed extract, which is about 50% phenol, in other words, about as pure as you can get it by initial preparation, is only 25 milligrams per liter. So the minimum threshold at 25 milligrams per liter, and then if we fractionate that extract into different components, the tannin is only 3.5 milligrams per liter as a minimum threshold. The leuco fraction about 20, and the catechin fraction about the same. The tannin fraction in this case is the larger molecules which are not removed from a chromatographic partition column by ethyl acetate. The leuco compounds are those which are removed by ethyl acetate after the removal of the catechin fraction with ether on a partition column. So partitioning with ether brings off the catechin, with ethyl acetate brought off the leucose, and the tannins were everything else in this case. Now some more up-to-date work is being done by Mr. Arnold, and you'll hear about that sometime in the future, where an additional fraction using butanol has been made of the presently collected group of condensed tannins. We have shown that there is a molecular weight difference, however, and the tannin fraction is larger in molecular weight than the leuco fraction, and the catechin fraction is monomeric, at least as far as the identified components in it. The difference threshold in white wine that contained 170 milligrams per liter of gallic acid equivalent, in other words, a fairly light white wine, were these figures. So the addition of the whole extract, it only took 80 milligrams per liter to make a detectable difference in the wine. And this difference was equally detectable whether decreased acidity was noted If you add tannin, you get a more milder flavor, and it tends to cover up acid, at least at the threshold level. I think it tends to interact at the higher level, but that's another story. And bitterness, if you ask people to rate for bitterness, you got the same threshold. If you ask them to rate for astringency, you got the same threshold. With glucose, you got about the same picture, but more effect. No, excuse me, with the tannin rather than the whole extract, you got more sensitivity, again, as you would predict from the water test. Only 12 milligrams per liter affected acidity and bitterness recognition, but it took a little more for the astringency. Glucose was much less potent and affected the acid less than it did the bitterness or the astringency. Catechin affected the acid more than it contributed bitterness, and it did not produce astringency at any reasonable level. So the general picture would be that catechin could, and its related compounds could affect bitterness, at least in red wines and possibly in some white wines under the natural circumstances, but would not affect astringency, whereas the smaller tannin-type molecules of a glucose sort would affect bitterness and perhaps astringency, but it tended to be present in so little amount that the tannins of the condensed type would contribute much more astringency and possibly also more bitterness. We have other data that indicates that the bigger molecules tend not to be so bitter if you get them sufficiently cleaned up, and the bitterness tends to reside in the smaller molecules to some extent. Pumice contact in later, or the most recent work, this is not yet published but has been submitted, if we made white wines with pumice contact sufficient that the total phenol raised only 100 milligrams per liter, we did also get then an increased rating, a significant increase in rating for astringency. So pumice contact equivalent to give 100 milligrams per liter of additional phenol did give additional astringency. It did not necessarily give increased bitterness, and there seems to be an antagonism between the two, so that if stringency is high, then the bitterness is not so objectionable. And again, I believe that we just had inferential data from our findings. I believe better data will be coming out shortly on the point. Well, how do these compounds act bitter? They act bitter by hydrogen bonding, as we said, and if we look at some typical structures of protein, and taking gelatin as an example of protein, gelatin is made from collagen, which is a three-stranded fiber, which is then unrolled, and the individual fibers become gelatin. And an individual fiber would be a polymer which contains a high amount of glycine, proline, and hydroxyproline, and in fact about 80% of the total amino acid is due to those three amino acids. So it would look about like this. Now this is a somewhat flexible chain, and it could bend around and complex with the tannin, so that it would tend to precipitate tannin just as would any other protein, but being fairly small, single chain, and flexible, it could snuggle up to, if you like, tannins better than some other proteins, and would do a better job of precipitating than less soluble proteins. Polyglycine would be a more regular appearing polymer, and we would expect it to have the most affinity of any protein, and it's sort of a protein, because it would have the most carbonyl compounds, the most amide groups. It's the smallest amino acid. Therefore, it would have the most frequent possibilities of hydrogen bonding with a phenolic OH. Nylon 66, for example, can bond with itself, as for that matter, so could polyglycine. And if it's bonded with itself, it might not be such a good precipitant. But when sufficiently separated or on the external faces of the bonded areas, it would be a good hydrogen bonder, but much less frequent ketone groups to serve as hydrogen bond acceptors. Of course, the nitrogen groups can be hydrogen bond donors, and that might have a bearing if some of the phenol is oxidized to the carbonyl form. So these kinds of materials would remove both unoxidized and oxidized phenols by similar mechanisms, but depending on the nitrogen group to donate hydrogen if it's taking out oxidized quinone type material, and depending on the phenolic hydrogen donor with the carbonyl if it's taking out normal unoxidized phenol. Polyvinyl pyrrolidone, PVP, has this kind of structure, and again, it's kind of hard to see these are carbonyls, but they are, and they have some flexibility. And since the nitrogen has no free hydrogen on it, it wouldn't be a hydrogen donor. So that's one reason that PVP tends to have a much higher affinity on a weight basis for phenols than does nylon. And these are, I know that's a little hard to see, but I hope you can see it. If you care to, if you can't, well, you can see it later. If we look at gelatin complexing at different levels in wine-like model solutions, you'll see that the gelatin will take out up to slightly more than its own weight. So if you added 100 grams of gelatin, you might take out 126 grams of polymeric tannin if you had a low level of gelatin. As you raise the level of gelatin, you'll take out less and less. And of course, if you had an excess of gelatin, it wouldn't all precipitate. The precipitate would still have maybe 60% tannin in the gelatin. So that generally speaking, this is about a mole for mole, or since they're both large moles, equal weight precipitation. In the case of nylon and polyclar, it's much less than an equal weight, and these figures were obtained by column treatment where we got complete saturation, but only 2% instead of 100% of the weight, up to maybe as much as 25% of the weight could be complex, so that they're much more selective than are the casein or gelatin types of proteins, but nevertheless they do about the same kind of thing by the same sort of mechanism, and that's why these compounds are astringent. Now that's about all I can tell you about the wines, but say around a half a gram of wood contributes enough phenol that you can make a threshold difference in wine. Now this threshold difference is partly oak odor, which may not be phenolic, but in any case it does indicate that about a half a gram of wood, which contains about 30 milligrams of phenol, will give a tasteable difference when that extract is added to wine. Our time, I think, is now up, and I believe this covers the main things I wanted to bring up about wine.